Hello friends, welcome to Sam Biology class. Friends, today we shall discuss about the cell wall of gram-negative bacteria. As compared to the cell wall of gram-positive bacteria, the gram-negative cell wall is quite complex. It has a 2 to 7 nanometer thick peptidoglycan layer covered by an 7 to 8 nanometer thick outer membrane. This is the peptidoglycan layer, which is 2 to 7 nanometer thick, and this peptidoglycan layer is covered by a outer membrane which is 7 to 8 nanometer thick. Because of the thicker peptidoglycan layer, the cell wall of gram-positive bacteria is more resistant to osmotic pressure than that of gram-negative bacteria. All the structures from plasma membrane outwards, including plasma membrane, cell wall and capsule, if present together, called as cell envelope. When examined with electron microscope in gram-negative bacteria, a space is seen between plasma membrane and the outer membrane. This is the space that is present between plasma membrane and the outer membrane. This space is called as periplasmic space. The periplasmic space is filled with a substance called as periplasm. The composition of periplasm in gram-negative bacteria is different from that of gram-positive bacteria. Sometime in gram-positive bacteria also, periplasm space is seen between plasma membrane and the cell wall. So, in gram-negative bacteria, the peptidoglycan layer lies outside the plasma membrane and is bounded on either side by periplasmic space. The width of the periplasmic space in gram-negative bacteria ranges from 1 nanometer to as great as 71 nanometer. In case of E. coli, the width of the periplasmic space is 2 nanometer and it contains 1 or 2 sheets of peptidoglycan. The composition of the periplasm in gram-negative bacteria is strikingly different from that of gram-positive cells. The periplasm in gram-negative cells contain a variety of proteins and enzymes that are involved in various activities. Some of the periplasmic proteins are involved in nutrient acquisition, for example, hydrolytic enzymes and transport proteins. Other periplasmic proteins are involved in energy conservation. Some are involved in peptidoglycan synthesis, and some of the periplasmic proteins even modify toxic compounds that could be harmful to the bacterial cell. One of the most abundant protein in the outer membrane is Brown's lipoprotein, and this is the outer membrane which is composed of lipids and proteins. The most abundant protein in the outer membrane is Brown's lipoproteins, and the outer membrane is connected to the cells by two different mechanisms. One is the outer membrane is connected to the underlying peptidoglycan by means of Brown's lipoprotein. These are the molecules of Brown's lipoprotein through which the outer membrane is connected to the underlying peptidoglycan. The second mechanism is that there are specific adhesion sites which are present on the outer membrane as well as on the plasma membrane, which are in direct contact with each other. So because of the presence of adhesion sites, both on the outer membrane and the plasma membrane. This membrane is connected to the cells. Friends, this figure shows the gram-negative envelope. The term envelope includes the plasma membrane and all those structures that are present outside it. This is the plasma membrane. Outside the plasma membrane, there is a thin peptidoglycan layer. This peptidoglycan layer is bounded on either side by periplasmic space. This space is filled with periplasm. Outside the periplasmic space, this is the outer membrane, which is structurally similar to plasma membrane and is composed of a phospholipid bilayer in which various protein molecules are embedded. Possibly the most unusual component of outer membrane is lipopolysaccharides. Lipopolysaccharides, also known as LPS, are complex and is composed of lipid and carbohydrate. A lipopolysaccharide molecule has three parts. They are the lipid A, the core polysaccharide, and the O-side chain. The fatty acid chains of lipid A are embedded in the outer membrane, and the remaining part that include the core polysaccharide and the O-side chain project from the surface. Lipopolysaccharide play a variety of roles. It helps stabilizing the outer membrane. It acts as a permeability barrier. It also promotes the attachment of bacteria to the surface and biofilm formation. Because the core polysaccharide contains charged sugar and phosphate group, because of this, the lipopolysaccharide also contribute to the negative charge on the surface of gram-negative bacteria. In addition, the lipopolysaccharide also protect the bacteria from host defense. One of the important components of lipopolysaccharide is the lipid A. The lipid A portion of LPS is toxic, so lipopolysaccharide act as an endotoxin and is responsible for some of the symptoms that are seen in gram-negative bacterial infections. When lipopolysaccharide or lipid A enter into the bloodstream, this leads to the development of a form of septic shock. The terminal part of lipopolysaccharide is O-side chain, which is also known as O antigen because of its antigenic nature. The O-side chain elicits immune response that leads to the production of specific antibodies. However, some of the pathogenic gram-negative bacteria rapidly change its antigenic nature, thereby escaping from the host immune system. Despite the role of lipopolysaccharide as a permeability barrier, the outer membrane is more permeable than plasma membrane and allow the passage of glucose and other monosaccharides. This increased permeability of outer membrane is due to the presence of a specific protein called as porins. So porine molecules cluster together to form trimers. These are the porine trimers. Porine molecule is tube-shaped and form a narrow channel which allow the passage of substances which are not larger than 6 to 700 Daltons. So friends, this is all about the structure of gram-negative cell wall. For more updates, please subscribe Sam Biology class and also please share your comments. Thank you very much. Good night.